we may be more familiar with species that invade islands and devastate the ecosystems, such as the brown tree snake, rosy wolf snail, rats, the list goes on. But introductions of species from islands to the mainland also occurs. From a physiological standpoint, the introduction of species to a new environment poses challenges with exposure to novel conditions. Our focus here is new habitat and thermal conditions. While many islands have admittedly complex habitats, they're geographically constrained and smaller area compared with the mainland. Ectotherms are more sensitive to thermal environments, and so they face additional challenges with thermal tolerance and performance in new ranges. The movement or introduction of an island species to the mainland provides opportunity to investigate the challenges and responses of a species to a new environment, so that we may better predict plasticity's involvement in establishment success for practical management reasons, such as invasive species risk assessment or repatriation programs. It also helps us to understand evolutionary processes and selective pressures behind successful range expansion. We used a lizard native to islands to investigate the utility of environmental and physiological tools to predict its range expansion onto the mainland. The northern curly-tailed lizard is a native to the Bahamas and Cuba and has established multiple populations in Florida, including some outside the latitude of its native range. Similar studies have been conducted on different knoll species, which demonstrate acclimation and adaptation to new thermal environments. But knolls are toy dwellers, and they lack behavioral thermoregulatory buffers, and thus might not be representative of other ectotherms, such as the curly-tailed lizard, which will burrow in extreme temperatures. No thermal physiological information existed for this species before our study, making it a good case study to objectively evaluate the thermal influences on a mainland invasion. Now, several hypotheses could explain the broad distribution of the curly-tailed lizard in Florida. The first being thermal matching. The distribution of the lizard, displayed here as white dots, is constrained by thermal conditions and associated tolerance present in the island native range, such that the lizard only occurs in areas matching the thermal conditions of that native range, and these matches are shown here as lighter shading. A second hypothesis is thermal potential, where the distribution in the native range is constrained by geographic barriers such as seawater, not thermal tolerance. In essence, the thermal tolerance of the native population exceeds what is needed for that native range, such that the lizards can survive in areas not found in the restricted native range. Lastly, thermal plasticity may explain the distribution. Physiological acclimation and/or adaptation both occur potentially in these novel environments, such that improved thermal tolerance is seen in areas outside of those thermal matches of the native range. These improvements are represented here as different shading. To distinguish between the thermal matching and thermal potential hypotheses, we constructed ecological niche models from climate variables from native range presence points, which were thinned to prevent spatial autocorrelation. The variables that we used after removing those that were correlated and to prioritize our hypothesis testing were mean diurnal range, precipitation seasonality, precipitation of wettest and driest quarter, and minimum land surface temperature. These values were restricted from the 1990s onward, after which most populations had established. We used ENM eval to select the best model from a selection of combinations listed here. Our model then predicts a suitable area based on the native range information, and so the suitable area is shown here as a lighter color. We'd expect that if thermal matching is supported, then our species should not really occur outside these high suitability areas. We can apply confirmed established populations to our map from published accounts, museum records, and confirmation by us in previous studies to assess whether they occur outside these areas. If thermal matching is rejected, we expect to see populations in low suitability areas as predicted by our model. So we did just that. And here we have a gradient of low suitability in lighter colors to high suitability in darker colors. The established populations are shown as white dots, and they occur within many of these light-colored areas. Our model thus dismisses the thermal matching hypothesis. This is largely where the model loses utility. Now, it's important to remember that this is an indirect way of evaluating thermal tolerance. In essence, it makes a lot of assumptions based on presence points, but often physiological data is lacking for most species, especially new or suspected invaders. So this is often the only tool available to assess the potential distributions. To distinguish between thermal potential and plasticity for our species, we needed to have some physiological tolerance information. So to test between these, we evaluated thermal limits in 35 lizards at each of 2 locations in March 2020. Both of these populations are coastal and have been established since the 90s, one in a low suitability area, as predicted by the ENM, and the other in a high suitability area well within the native range. All these lizards were collected in a span of 7 days. We assessed the thermal limits on the same day of capture, and we evaluated CT min before CT max, allowing all lizards to have a recovery period going through all CT mins before CT maxes. We used Gators, which is a device which manipulates environmental chamber temperature at a 1 degree Celsius ramp rate and records lizard body temperatures with a thermocouple that's inserted into the cloaca. The chambers are independently movable without touching the lizard, so we're able to assess the loss of riding response with minimal change to chamber or lizard temperature. We evaluated this data using ANCOVA with the main affected population and covariates of mass and sex. We predicted that if potential was supported, we would observe no differences in thermal limits, but if plasticity was, we would observe some differences. And we found both. There was evidence for the thermal plasticity hypothesis in the critical thermal minima, as the northern population showed 1 degree Celsius lower CT min than the southern population. There was no detectable difference in critical thermal maxima, so we can't distinguish between thermal potential or plasticity, although it is possible that plasticity in this CT max occurred similarly after introduction in both populations. Now, the CT min appears more evolutionarily labile than CT max, according to most of the literature, in that the CT min is not as close to the lethal minima, and so it has more potential to be plastic. Looking at the difference in the CT min, it's difficult to distinguish between acclimation and adaptation effects. Displayed here are the ambient temperatures for both populations leading up to the sampling, minimum in blue and maximum in red, solid lines and X's being the northern population. The blue dotted line shows the average CT min from the study. There were several instances where the temperatures dropped below the CT min, but a notable cold snap occurred in both populations about 40 days prior to sampling. I circled it here. A very recent study in tree-dwelling lizards found this same cold snap influenced CT min up to 10 weeks afterwards in several species in Miami. However, the snap affected both locations of our study, so probably can't explain the CT min difference between our populations. Another recent study found no evidence of seasonal acclimation of CT min in our study species in an intermediate population. This was published after we had collected all of our data. But we hesitate to draw conclusions because it is unknown how long these lizards spent in captivity before this testing. Regardless, it's important to note that burrowing lizards can buffer short-term extreme temperatures better than tree-dwelling ones, so a short-term cold snap might not affect them as much as longer-term. And looking at the longer-term thermal environment of our two populations, it's apparent that while the maximum temperatures differ only slightly, the minimum temperatures are lower in the northern population, and average minimums fall below the CT min for 4 months of the year in the north, yet remain above this threshold in the south. So there seems to be regular selective pressure for better cold tolerance in the northern population, which may explain the lower CT min. Combining our physiological data with an improved niche model that includes the established non-native populations, we find that land surface temperature thresholded at the CT min, which accounts for some burrow buffering, is shown here as diagonal lines, pretty well approximates the predictions of our best correlative ENA model with foresight. Notably, in other cases, we would not have this data of already established populations because in most cases, these models are attempting to predict onto a new area. Now, both fall short, and mechanistic niche models may fill that gap and provide better insight, but are more complicated and difficult to collect all that information. So it is possible that relatively simple and quick thermal tolerance info may be comparable to range predictions for range-restricted species, or where distribution information is lacking. Thank you for listening, and I'll take any questions.